Hello students, as you can see I am going to start the next point, evidences for DNA as a genetic material. So here the first evidence is given, which is the Griffith's experiment. Frederick Griffith in the year 1928 performed an experiment. He used two bacterial strains of pneumococcus, also known as Diplococcus pneumoniae. These two strains were R2 and S3. R2 means a rough strain which produced rough colonies, and the cells of these colonies were without any capsule, while the S3, that is smooth strain of same Diplococcus pneumoniae, was producing smooth colonies because the cells were having a capsule surrounding the cell. So let's see how the Griffith used it. So as you can see in the Frederick Griffith's experiment, he performed 4 steps using the R2 rough strain of Diplococcus pneumoniae and S3 smooth strain of Diplococcus pneumoniae. So in the first step, he took S3 strain suspension in a syringe, injected into living mice. The mice died because the S3 cells were capsular cells and the capsule is having D amino acid, which is very lethal for the immune system of mice. So due to the capsular activity, the phagocytic cells were unable to kill S3 and the S3 infected the mice, due to which mice died. While in the second step, Griffith performed the experiment by using living R2 strain injection of suspension injected into living mice, where the mice lived. Why mice lived? Because R2 cells were killed by phagocytic cells of the immune system of mice, because they did not contain capsule which is made up of D amino acids. So in general, what happened in these two steps, when S3 is injected into living mice, living S3 injected into living mice, the mice died, while the living R2 when injected into living mice, the mice lived. Means the S3 strain was pathogenic to mice and R2 strain was non-pathogenic to mice. So let's see about the next steps, step number 3 and 4. As you can see students, in the third step, Griffith performed the same step but with heat killed S3, not the living S3. So when heat killed S3 suspension is injected into living mice, the mice lived because the S3 cells were killed before injecting by boiling. In the fourth step, the same heat killed S3 suspension is taken along with living R2 cells. So when it is injected into another living mice, what happened? Surprisingly, the mice died. The fourth step, mice died. How it was possible? Griffith confused. So he again observed the S3 cells were, yes, completely killed, 100% killed. But something from these dead S3 cells transformed these R2 into capsular S3. So let's see in the diagram how it happened. Hello students, as you can see in the diagram, the heat killed S3 cells were mixed with living R2 cells, no doubt. All heat killed S3 cells were confirmed that they are killed. Then also something from dead S3 cells was taken up by R2 living cells, which was one extra character from the S3 cells, that is capsule character. So these living R2 took the capsule character from heat killed cells S3 and converted into living S3. So living R2 got converted into or transformed into living S3. So this transformation occurred. He means who? Mr. Frederick Griffith named this transformation substance as transforming principle. He did not know at that time it was carbohydrate, protein or DNA or RNA or lipid. So he got confused and named it as transforming principle. We will see in the next video the second evidence for DNA as a genetic material. Till then, bye bye.